Technology is everywhere and is, of course, changing our life in many ways, trying to make it easy. The medicine and the illness treatments are one of those items in our life which are under permanent change because of technology. Physicians need of elements that allow us to see how illness are progressing in our bodies or even if we're healed. This is achieved by means of a little device named sensors that are able to speak with us about our bodies, fields, and so answer very simple questions such as, is your body cold? Is your heart beating too fast? Is your body infected for any bug? In this exhibition, you will learn how sensors work and how the modern technology can learn from nature. Our bodies are very wise, and they can fabricate large molecules named proteins, acting as natural sensors. For example, they make us feel whether through touch of something is hot or cold. Let's see in the present exhibition how the proteins could give us the possibility to build new sensor bio-inspired, thus being closer to nature.
Hello everyone! Have you ever wondered how computer simulations can help us understand and design the sensing part of our biosensors? Today I'm excited to take you on a journey into the world of in silico protein engineering and molecular modeling in the context of biodegradable biosensor development. Imagine being able to see the inner workings of molecules without a microscope. This is where computer simulations come into play. Through sophisticated simulations, we can visualize and manipulate molecules, providing invaluable insights for designing biosensors. Molecular modeling allows us to create 3D structures and representations of proteins and simulate their behavior. It's like having a digital laboratory at our disposal, which is particularly useful for crafting biosensors. Ion channels found in our bodies are natural sensors with great potential for biocompatible biosensors. However, to harness their power, we need specific mutants tailored to meet certain sensor criteria. Molecular modeling and in silico protein engineering help us to identify the structural modifications that can provide the required sensing parameters and properties, making ion channels the ideal sensing elements for our biosensors. In conclusion, computer simulations and molecular modeling techniques are insightful companions of our biosensor development journey. They empower us in designing innovative biodegradable biosensors, with ion channels as the ideal biocompatible sensing elements. These biosensors have the potential to have a significant impact on healthcare and medical monitoring.
Hello everyone. Today I want to talk about one of the most fascinating parts of our biology. How do we sense temperature? So if I hold this cup of coffee in my hand, I would instantly know if it's hot or cold. But how is it? Let's talk about the science behind it. So inside of our body, we have these small detectors that are called TRP ion channels. These ion channels will get activated with change of temperature, and they will send signals to our brains so we can perceive the temperature. But one of the most fascinating things about these channels is that they won't just get activated by temperature. I want to give you an example. Have you noticed that when you are chewing a mint gum, you would instantly feel cold? It's because a member of this ion channel family, TRPM8, which is a chill detector, will also get activated by menthol. Or another example, have you noticed when you are eating spicy food you would instantly feel hot? It's because TRPV1, which is a hot detector, will also get activated by capsaicin in the spicy food. These channels are essential for our body. We need our body to stay in the same range of temperature, and regulating this temperature is essential for keeping the enzymes functional and keeping our cells healthy. Now imagine how much cool it would be if we could use this naturally occurring sensor in biomedical applications. Imagine if we were able to build a sensor based on these thermotherapies. So one day we would be able to detect the smallest change of temperature in our body, which could be a sign of infection or other things that could go wrong. It's truly amazing how much we can learn from nature, and it's even more exciting trying to put that into applications.
Hello everyone, my name is Hanna and I'm PhD student at University of Lausanne, Switzerland. Today we are going to talk about fascinating aspect of our body physiology, which is the control of body pH. Did you know that our body maintains a delicate balance of acidity and alkalinity which is crucial for our health and well-being? Firstly, let's understand why pH is important. pH, or potential of hydrogen, is the measure of the acidity or alkalinity of a solution. In our body, maintaining the right pH level is vital for various biochemical processes to function optimally, including enzyme activity, cellular metabolism, and the function of several organs and systems. Now, let's delve into the role of acid-sensing ion channel, or ASICs. In our body, ASICs are specialized ion channels found in neurons and other cells through the body. These channels act as natural pH sensors, detecting the change in the surrounding environment. When the pH of the cell environment changes, ASIC respond by opening, allowing ions such as sodium or calcium to flow into the cell. This process triggers a cellular response, which can range from pain sensation to neural signaling. So, what is the significance of understanding ASICs and body pH? Well, we are exploring the potential of optimizing ASICs properties to create natural implantable biodegradable biosensors. This biosensor could be used for various applications and can be used for non-invasive, fast, and real-time monitoring for body pH. By harnessing the properties of natural ion channels like ASICs, we could develop innovative solutions for healthcare professionals and decrease the invasive diagnostic procedures. Thank you for watching my video, and I hope you found it informative and insightful.
Hi, I am Alexandrine Kurtz, the ESR4 in the BioInspireSensing project. I'm based in the University of Teramo, Italy. So, the BioInspireSensing project aims to take advantage of nature in order to create a bioresorbable nano device able to monitor from the inside of the human body physiological parameters such as temperature, pH, and cholesterol. Proteins being naturally produced by organisms, using them for sensing would allow a bioresorbable feature of the device. In the framework of temperature sensing, two proteins have been selected, and in order to produce them, the DNA sequences encoding for these proteins are injected into cells that will be able to produce these proteins, from which the proteins will be isolated, extracted. To achieve the final goal of temperature sensing, we need these proteins to retain their activity after purification. Afterwards, the proteins will be embedded into membranes that will be set up on the electronic device for signal detection.
Hi everybody, I'm Caterina Brisutti, the ESR number 5. I'm currently doing my PhD in Groningen in the Netherlands, and my project consists in studying the biophysical and the structural properties of hybrid membranes. Membranes made of lipids, so the native component of the biological membranes, and amphiphilic block copolymers. molecules that provides more stability into the membranes. And in addition to this, we also want to study the mechanism behind the activation of complex membrane proteins that need to be embedded in a specific lipid composition. So we want to combine biosensing membrane proteins and more stable platform in order to have a biosensor as much as possible stable and effective.
Welcome to the exciting world of biosensors. Today, I'm going to take you on a journey where building a biosensor is like constructing a room. Like in every room, we need basic building blocks called bricks. In the scientific sense, these bricks are going to be polymers and lipids. Like how you join the bricks together to prepare a floor, we are going to use membranes prepared from polymers as our base and the foundation for our sensor. For the roof that protects us from unwanted elements entering into our room, we are going to use lipid bilayers. So now that we have a floor and the roof, we need to stick them together using pillars, or also called as anchor lipids. These are going to provide us with an open space for the ions to move freely, and also providing us with structural integrity and support for our biosensor. We have now the basic structure of the room, but it's not fully functional. We need to implant that with functionalities using proteins and biomolecules that gives us unique capabilities to our biosensor and also its purposes. So to conclude, in the world of biosensor, creating a biosensor is like constructing a room. We use building blocks like polymers and lipids, creating roofs and floors and sticking them together with anchor lipids. However, it is the proteins and biomolecules that provides us with unique functionalities and capabilities by transforming our biosensor into a fully decorated room of discovery that are ready to analyze and detect the world of molecules and ions around us. Thank you.
Do you often wonder what's happening inside of our bodies? Sometimes our body tells exactly what it needs. For example, on a hot day, our body needs more water, so it sends signals to our brain. We feel thirsty and we drink a glass of water. Same thing if the body needs more energy, it sends signals to the brain and we feel hungry and we eat a sandwich. So it's very important to communicate with our bodies because this tells us if we're still healthy or if we're developing some sort of disease. Communicating with our bodies can be done through sensors. These sensors can be like those COVID test strips that we used during the pandemic, or they can be our smartwatches that monitor our heart rate or blood oxygen levels. And these sensors can also be placed inside the body to know exactly what's going on. So, all of these sensors have one thing in common, and that is the electrode. The electrode acts as a bridge between the body and also the computer that analyzes all of these signals. So, in our BioInspireSensing project, my project is to develop these electrodes.
Nature is incomprehensibly beautiful with endless power of magic and wonder, so it's logical to take advantage of it to have better life. Developing biomimetic bilayer lipid membranes for sensing applications has played an essential role in recent years, since it can pave the way for tremendous applications such as diagnosis of disease and monitoring cellular responses. Moreover, it facilitates development of novel biosensors which are able to detect different physicochemical factors. The possibility of this fact lies in our project. In my PhD thesis, we are trying to engineer and develop different and various biomimetic bilayer lipid membranes, including liposomes, vesicles, and bicells, by taking advantage of natural cell membrane composition, which is mainly lipids and proteins. So, by incorporating ion channels inside these lipid membranes, we are able to detect pH, temperature, and pressure changes in our cells. So, as a result of this project and this research, we are able to create a great impact in biosensing field.
How do we test our device without using animals? To answer this question, we first need to understand why we cannot keep using animal models anymore. Besides the obvious ethical issues, animal bodies are terrible at predicting how human bodies will respond to medicine. For example, in 2018, 90% of the drug that showed promising results in animal models failed clinical trials. Additionally, the time for a newly developed drug to enter the market could take as long as 15 years, and finally, the development costs could be as high as up to $2.6 billion. This makes the use of animal models unreliable, slow, and expensive. What else can we use to test new drugs or materials? Researchers are looking into ways to make animal models redundant. One solution could be the use of microphysiological systems. These are small environments to which we can add human cells, and due to the nature of these environments, the cells can form unique structures mimicking a small part of the physiology of our own bodies. These microphysiological systems hold many advantages. For example, they are designed to mimic human organ function and structure, making them more relevant for studying human physiology. Also, we can control environmental conditions such as temperature, oxygen level, and fluid flow, leading to increased experimental reproducibility. And finally, the ability for high throughput screening, because these systems can be miniaturized and automated, enabling high throughput screening of compounds, which is valuable for drug discovery and toxicology studies. Then finally, how can we use these environments to test our device? One way of testing our device is by adding it to our specific microenvironment and use microscopy to observe how it responds and when we expose it to different stimuli. This way we can see what the behavior of our device is to these stimuli and when it's surrounded by an environment that is much more like the human body than an animal body is. Hopefully we can use these results to anticipate future issues and to test biocompatibility.
Now that we know a little bit more about biosensor, let's say how we test it for our final scope. First of all, we have to think about which biological component we want to sense and detect and which recognition element we will use for it. First of all, we do an in silico analysis. That means via computational methods and theoretical models, simulate the structure of this receptor and see how it interacts with the analyte. Then we move to in vitro assays, what literal means in glass, and we do this to test and evaluate the biosensor outside living organisms. A biosensor as an analytical device can be tested with different techniques like microscopy with cells, optical fluorescence, electrochemistry, or even mechanical assays. In vitro assays are a crucial step on the development of a biosensor because we can use it for optimize and validate the biosensor in biological systems.
Hello everyone, my name is Isam Kuri Avinahur. I am ESR 11, responsible of developing the doctoral thesis, which is called Proof of Concept Electronics for In Vivo Testing. In this poster, here I present you some main points to highlight about the BioInspireSensing Consortium and what we are going to do with the animal testing after presenting and developing and testing all the electronics. So, as I told you, in this diagram, we will present 3 points that I consider important and will resume very well my main goal inside the consortium with my project. So, the first point is the design and fabrication of the electronics that are responsible to capture or to record the signals that are coming from the dynamics of the selected ionic channels that the other students tested before. The circuits will be responsible for injecting current and monitor the voltage variations that are coming from these ionic channels after perturbations of pH or temperature. Those ones are external perturbations. Question that might arise from this point is that how big or small these electronics should be. Also in this point that they should be wireless or not. After we got the signals, we should work on some signal processing, which is a parameter extraction of some values that in this case will be the innovative points, because then it can be used later as a reference to test these ionic channels and to replicate them into circuits and see if the nature and the imitation behaves very well. And lastly, for the animal testing, will be provided by a specific rat with a specific mutation that can alter its ionic channels. If it's not achieved in this point, we can do this recording device for in vivo testing, on which we can use it for muscle cells, for artificial ionic channels, or some ionic channels that were isolated before, or with some types of cells that I have been developed during the work of my master thesis. Later, and after so many trials, it can be enhanced and used for proposals and for monitoring diseases in a human being. Thank you so much for your attention into the topic.